This is the video animated version of the Radwa and Radwa podcast, where we aim to communicate scientific research to science enthusiasts. We post 4 versions of each episode, in English and Arabic, and in podcast and YouTube video formats. All the links are in the description below. You will also find our references in the description. We are your hosts, Radwa Ewaisha
and Radwa Sharaf,
PhDs, pharmacists, and molecular biologists. In our episode today, we will be talking about PCR. So many people started to talk about PCR more when COVID hit. And today we would like to ask Radwa to explain to us what the PCR technique is.
Sure. So PCR is short for polymerase chain reaction. And it's a technique that we use in the lab in order to synthesize thousands, if not millions of copies of a specific DNA that we're interested in detecting. You can think of DNA as a fingerprint. So if it's present in our sample, that means that the organism from which this DNA originated was also present in the sample. So if we're trying to detect the presence of a specific virus, we can look for its DNA using PCR. We can also use PCR to detect bacteria and synthesize copies of its DNA, but also we can use it to multiply the DNA present in our human cells. So for example, if we have a sample from a scene of a crime, we can use PCR to generate millions of copies of that DNA.
So why would we be interested in generating millions of copies of that stretch of DNA?
Yeah, so usually the sample that we obtain has a small quantity of cells, and inside these cells we have the DNA. So if you remember when we spoke in our episode about DNA, we said that it is found inside the nucleus of a cell. However, when we try to extract that DNA, if its quantity is low, first of all, it may degrade because we manipulate it in the lab, and we may be unable to detect that DNA moving forward. And experimentally, our tests and experiments do rely on the presence of large quantities of DNA in order to allow for proper detection.
Okay, so can we get a little bit more technical? How do we extract DNA from the sample?
So let's think about the different types of samples we may obtain. We could have a blood sample, we can have a swab that has some respiratory secretions, we can have a saliva sample, a sample from skin or hair or stool. All of these represent samples that contain cells from which we can try to extract the DNA. We said DNA is present inside the nucleus, inside the cell, and both the nucleus and the cell are surrounded by a membrane that's made up of a lipid. It's a fatty layer. So what can we use in order to dissolve this fatty layer and expose the DNA that's present inside of it? It's just like the same concept of using soap. We use soap in order to break down fats present on the dishes. And in the lab, we can use chemicals that have this same surfactant property that can dissolve this fatty layer surrounding both the nucleus and the cell in order to extract the DNA present inside.
Okay, so now we have the DNA. How do we set up a PCR reaction?
A PCR reaction is composed of 4 primary components that are necessary for the reaction. The first component is the enzyme, also known as polymerase, from which the name polymerase chain reaction, PCR, originated. And this enzyme catalyzes the synthesis of millions of copies of DNA from our source material. This enzyme has to be kept at a very specific temperature. Certain conditions need to be maintained in order to allow for the catalysis process, and it has to be present in our reaction in order to allow for the PCR to proceed. The second component are the units that will be used to synthesize these millions of copies of DNA. So you can think of these millions of copies of DNA as a skyscraper. In order to build this skyscraper, we have to obtain the building blocks and the units needed to synthesize and form each one of the different floors on this skyscraper. And these building blocks are called DNTPs, deoxyribonucleotide triphosphates, short DNTPs. They're used to generate and form these copies of DNA that we're interested in generating. The third component that's necessary for the reaction to proceed is what's known as a primer. So a primer is a short sequence of DNA that is used as a starting step for the synthesis of DNA. And this primer is essentially what dictates which DNA species we will be propagating and synthesizing more copies of. So if we're interested in SARS-CoV-2, the virus that results in COVID, we'll be using a primer specific to SARS-CoV-2. If we're interested in multiplying copies of human DNA, we will be using a primer specific to the human sequence. And so the primer dictates the specificity of our PCR reaction. And then finally, the fourth component is a collection of reagents that are necessary to maintain a proper medium for our enzyme and our reaction to proceed. So this consists of a buffer that has specific ions like magnesium or manganese in order to help with the stability of the synthesized DNA molecules and allow for the enzyme to properly function. Once we've assembled all four components and added them to our sample from which we've already extracted the DNA, this reaction then needs to be incubated inside of a machine, also known as the PCR machine. Not very creative, I must admit, but that's okay. And this machine regulates the temperature at which the reaction proceeds. So specifically with PCR, the temperature does need to fluctuate. Sometimes the temperature needs to be maintained at a high level, like 95 degrees Celsius. Sometimes the temperature needs to be brought back down to 60 degrees Celsius, depending on the specifics of the reaction. But it does need to incubate in the machine for anywhere from an hour to an hour and a half, and sometimes it can go for as long as 6 hours. And once the reaction has completed, we will have amplified the DNA of interest if it's present in the sample. So let's think about the different possibilities. If the sample is positive, meaning it contains the DNA in which we're interested in, then the PCR reaction will result in millions of copies of that DNA, and then we will be able to easily detect the presence of that DNA. However, if the sample is negative and does not contain the DNA of interest, the PCR reaction will proceed, but nothing will get amplified. And so when we try to detect the presence of that DNA of interest, we will not be able to find it because the sample was originally negative.
Thanks, Radwa, for the clarification. So today we talked about PCR, which is polymerase chain reaction, and that is a technique that is used for the detection of nucleic acid from different organisms. So detection of nucleic acids is like detection of a fingerprint of that organism or virus. And Radwa talked about the specifics of a PCR reaction and what is included in that PCR reaction, and in the end, how to interpret results. So if it's positive, then this means that this specific nucleic acid is present and that organism is present.
We hope you enjoyed today's episode. Please don't forget to like and subscribe to our podcast and our YouTube channel. You can also find us on Twitter, Instagram, and Facebook. Feel free to reach out with suggestions for future episodes or leave us a comment directly on our YouTube video. See you next time!